Okay, hello to everybody. I wanted to thank you for joining us again on Friday for the Friday MindCORE seminar. We are joined this week, we're presenting the work of a new faculty member. Just a few housekeeping things I want to talk about is that you are welcome to post some kind of general comments or feedback or clap or appreciation in the chat. And if you have a question for our speaker, post it please in the Ask a Question section to the left. There's also an opportunity for you to upvote questions if you really like it. And we will be sure to try to get to as many of those questions that are top of the list when we can. So let us get started. I'm going to turn it over to Joe Cable, who is going to introduce our speaker today.
Great, thanks, Heather. So we're delighted today to be joined by Mike Arcaro, who's a new faculty member in the Department of Psychology. Mike did his graduate training at Princeton with Sabine Kastner and Greg Hassan, and did some great studies of the large-scale organization of the visual system, and then moved on to a postdoc with Margaret Livingston at Harvard, where he asked really foundational questions about, we know that the visual system consists of this tile of different areas with different sensitivities, and how much of that is present, from the get-go, and how much of that is dependent on an interplay with early experience. And I think the results that he's found, this is an age-old question in neuroscience and psychology that goes back to Hubel and Wiesel, and I think the results that Mike has found regarding the interplay between the interplay with experience over development in terms of the laying out the large-scale organization of the visual system are foundational, and we will be teaching them in our textbooks for many years to come. And so given that, we are absolutely delighted to have recruited him to attend the psychology department, and we're delighted to have him here today for the MindCORE talk to tell us about his work. And I will hand it over to Mike.
Thanks for that intro, Joe. I feel like you just did my intro for me. I could just skip right to the data. I'm very happy to be here and have this opportunity to talk about my lab's work with the broader Penn community. So as Joe nicely said in his introduction, my lab studies how the brain comes to represent our experiences and our environment, and how this in turn affects the way we perceive and interact with the world. And so I've been exploring this in the realm of object vision, specifically primate vision. Primates are very visual animals. Our visual perception is fast and accurate. We can effortlessly detect and classify objects in our environment in a fraction of a second. And that's despite the tremendous variation any individual object might have that can produce an image on our retina. So across individuals and species, the way we interact with our environment has a surprising consistency. So for example, many aspects of our visual perception behavior are comparable across individuals. As seen here up above, individual humans and chimps tend to look at similar salient features in natural scenes, such as other people, and especially the faces more than other body parts. And similarly, humans and monkeys perform comparably on complex object recognition tasks, so they tend to make similar errors in discriminating between different objects and get similar discriminations correctly. So data such as these really indicate that there's some common neural architecture that support these functions across individuals and across primate species. And it's likely that such behavior is not performed just by any individual brain region, but rather by a rich mosaic of probably anatomically interconnected, but functionally specialized regions. The central focus of my lab's research is on how these regions come to represent our environment and our experiences over development. So I approach this question by studying the visual system of primates. And one of the challenging aspects to identifying functions and organizing principles that are common across primates is that there's considerable anatomical variability in the size, shape, and folding patterns across individuals and especially across species. But despite this variability, there are several functionally specialized regions, particularly in inferior temporal cortex, illustrated over here, that support our visual behavior and our ability to recognize complex visual information. So over the course of this talk, you're going to see many of these types of brain images. So I just want to orient you to the macaque primate brain. These are inflated cortical surfaces of the left and right hemisphere. Back over here is posterior occipital cortex, where primary visual area V1 is. It's surrounded by extrastriate visual areas. Here's the intraparietal sulcus, where posterior parietal cortex is. This is the central sulcus, where primary somato-motor cortex is. This is the arcuate sulcus, so this is frontal and prefrontal cortex. And of main interest for today's talk is the superior temporal sulcus, and particularly the anterior half, where the ventral visual pathway and regions critical for object recognition are housed. And so individual regions within IT appear to be selected for particular important categories, such as faces, bodies, inanimate objects, and scenes. And pretty much all neurons within these regions are selectively active when an individual is viewing that image, such as faces, compared to any other complex shape, such as bodies, fruits, gadgets, so on and so forth. And we know that these clusters are important for normal visual behavior, as damage to these regions lead to specific deficits in recognition of that corresponding category. So damage to this red region, which corresponds to a face cluster, will lead to selective impairment of recognizing faces, but leave largely recognition of inanimate objects intact. So these clusters are found pretty much in all normal individuals, and they're located in stereotyped locations of IT cortex. And they've been shown to exist in several primate species, such as macaques and humans, and more recently in marmosets. This really suggests there must be some common influence guiding this organization across individuals and across species. So one of the prominent arguments is that the functions of these regions have emerged over the course of evolution and are innately predetermined. And that could explain this consistency seen across individuals and species. However, there are also specialized regions within the inferior temporal cortex that are involved in word processing over here in this green region. And these clusters are dependent on visual experience, specifically literacy. Also, monkeys, while they don't naturally read, can have text regions induced in young monkeys through intensive early visual experience and training. Interestingly, for both this visual word form area and these text regions, these clusters are located in stereotyped locations across individuals, similar to other regions such as faces and body regions. So even in these cases of experience-dependent brain regions, they can exhibit these properties of brain organization that are typically associated with arguments for a predetermined function. So despite decades of research on the function supporting object recognition in these areas within the IT cortex, how these regions come to support our visual perception has remained largely mysterious. And over the past several years, my lab has been researching these questions by studying the development of the cortical face network. And we're using the cortical face network as a proxy for really studying the development of high-level vision. And for today's talk, I'm going to review the main findings from some of our initial studies looking at the development of this face network, and then discuss some recent extensions of this work. So we know in adult macaques that IT cortex contains several functionally specialized regions that are selectively active when monkeys view faces compared to other types of complex shapes. So I'm showing you these regions here from fMRI experiment. They're in this red and orange region. And so we know that this organization is consistently found in adults, but up until a couple of years ago, we didn't know what this organization looked like in juveniles or newborn monkeys. So the first experiment we ran was to answer, are these face patches present at birth? So to do this, we scanned newborn monkeys starting just after 1 week of age, when they were robust enough to scan for an hour in an MRI. And then we scanned these monkeys longitudinally pretty much every week for the first 2 years of life. There's a whole lot of scanning. And to localize face regions, we use a very standard block design fMRI approach where we showed blocks of faces. So each face was presented for 500 milliseconds, and then we contrasted that with blocks of objects. And these are objects the monkeys are familiar with, that they have seen, they experience normally in their colony room. And we interleave these blocked presentations with fixation periods of no stimulation to get a good estimate of a baseline activity. And so just to remind you, based on studies in adults, we're expecting to see at some point face selective activity in IT cortex highlighted here in red. So by about 200 days of age in each of the monkeys we scanned, we found face selective regions in the stereotype locations of IT cortex. And you can see those here in red and yellow. And once these regions were identified in all subsequent scan sessions, they showed up. So here's 400 days. This is just shy of 800 days after birth. Prior to 200 days, though, we did not find face selective clusters in IT cortex. And that wasn't that the visual system wasn't responsive. IT responded to both the faces and the objects. There just wasn't any differential in the magnitude of the responses. So we took this to indicate that face patches are not present at birth. And that indicated to us that early visual experience is playing a critical role in their formation. But we also know that the visual system is immature at birth. The response properties of even V1 neurons are immature for the first several months. So it wasn't clear from these studies alone whether early visual experience is needed for the development of these face patches, or whether over the course of the natural development over the first several months, these face patches would just naturally emerge independent of experience. So the next experiment, we directly tested this by hand-raising monkeys for the first year of life without seeing faces. So every day we fed and we played with these monkeys several times a day. And we did so while wearing these welder's masks. So they could see our body, they can interact with our hands. They just couldn't see the details of our face, and they didn't see the faces of other monkeys. So here's a nice picture of one of the feeding sessions. Marge is feeding one of the baby monkeys. And here's a monkey that's a little bit older, and Peter is doing one of his playing sessions with that monkey that he does several times a day. So this takes a lot of effort to hand rear these monkeys. And during this first year, we scanned the monkeys several times, again, using a block design approach, where we looked at responses to faces, responses to objects, and we also looked at responses to hands. And the reason we looked at hand responses was because in control monkeys, it's also well established that in adults, there are hand selective regions within IT cortex, and they tend to be located in adjacent parts of IT cortex relative to the face selective regions. And again, this is all after 200 days after birth. So the big question is, what are we going to see in our face-deprived monkeys during this first year where they're reared without seeing faces? So across all the scan sessions in 3 different monkeys in this experiment, we didn't find any face selective clusters in IT cortex or any parts of the brain for that matter. However, in each scan session, we identified hand selective clusters in IT cortex in the approximate locations that we would expect based on adult studies. So this really nicely illustrates that this lack of early visual experience with faces had a selective effect on the development of face patches in IT cortex without a general effect of the development of IT cortex for other aspects of object recognition. In addition to the lack of these face patches, our monkeys raised without seeing faces also exhibited very different visual behavior. And we assessed this in a free viewing paradigm. So we showed all these types of natural images that had faces, and we monitored where monkeys looked in these images. And controlled monkeys spend the majority of time looking at faces in each of these images. In contrast, our face-deprived monkeys tend not to look at the faces, and rather they look at feet and especially hands in these images. So these are just 2 examples. We did many sessions in these face-deprived monkeys and the control monkeys. So each of these points is a unique free viewing session where they looked at many, many different images. The different symbol shapes are different monkeys. The white outlines are the cross-section averages for each of the monkeys. The y-axis here is fixation time spent looking at hands. That's contrasted on the x-axis, looking at fixation time spent looking at faces. And you can see that all the monkeys raised without seeing faces spent more time looking at hands than they did faces. And in contrast, the control monkeys, and this is consistent with several prior studies in both humans and monkeys, preferentially looked at faces in these natural images. So we took this as pretty strong evidence that visual experience is necessary for the development of these face patches. And this affects not only the organization of the brain, but also the behavior of these animals. And so this led us to wonder, well, how exactly are these face patches developing? How is this early experience influencing the refinement of circuits within IT cortex? And we had an early hint to this in the free viewing data, where we noticed that even within the first couple weeks after birth, these monkeys preferentially looked at faces compared to other aspects of these natural images. And that's well before that period where we first start to see face patches naturally emerge in these animals. So just to unpack that a little bit further, it's frozen. There we go. So these monkeys very early on are preferentially looking at faces. So that information is coming to their retina. Presumably that's getting relayed through the geniculate to V1, and then that propagates throughout visual cortex. And so the question is, how is that face information reaching these specific parts of IT cortex? And then over the course of the first couple months, that circuitry is refining to selectively respond to faces. And so one hint of what may be going on is that in adults, we know that the visual system is retinotopically organized. And that's to say that neighboring parts of cortex represent neighboring parts of, or receive information from adjacent parts of the retina, and thereby receive information from adjacent parts of visual space. We know that this organization exists in adults, and several studies in both humans and monkeys have established a link between this retinotopic organization and these category selective regions in IT cortex, such that foveal locations, illustrated here in red and orange, overlap with face selective regions in IT cortex. So very early on, if monkeys are fixating on faces in their natural environment, that face information is being selectively routed to foveal locations of the visual system. And if a retinotopic map is there early on, that could be a pathway for face information winding up in selective parts of IT cortex. So next we asked, well, are there retinotopic maps at birth? We know that the face patches aren't there until 6 months. What about the retinotopic organization? So we looked at our youngest monkeys. This is again looking at monkeys just over a week of age. Problem with doing retinotopic mapping in monkeys this young, though, is that these monkeys tend to spend a lot of time sleeping in the scanner. After about a month, maybe 3 weeks, these monkeys actually become really interested in the visual stimuli that we show in the scanner, and they stay awake. But during this first couple weeks, especially the first week, these monkeys spend a lot of time sleeping. And this makes it very challenging, if not impossible, to do retinotopic mapping, which requires monkeys to be trained to fixate. So instead of a conventional retinotopic mapping, we used a correlation mapping approach. And we implemented this by scanning the monkey, not just when the monkey was young, but also those same monkeys when they were older, and they could be trained to fixate so that we could get nice retinotopic mapping in those monkeys. And then we went back to the earlier week-old data, and looked at those fMRI sessions, and you could call that maybe spontaneous activity. It's not stimulus evoked, at least visual stimulus evoked activity. And we looked at whether the spatial pattern of temporal correlations reflected a retinotopic organization. So to put that more generally, we asked whether foveal regions of visual cortex were correlated with other foveal regions of cortex more than peripheral regions of visual cortex, and vice versa, were the peripheral regions more correlated with each other than foveal regions. And so we just categorized those spatial patterns of activity and clustered them in a color code to illustrate which regions were more correlated with each other. So these red regions were very correlated with each other. green regions were more correlated with each other, and blue regions were more correlated with each other. And so this revealed what looked to be a general topographic map that had features consistent with the eccentricity organization in the same monkeys when they were older. Specifically, this swath of lateral, occipital, and temporal cortex largely reflected foveal correlations. There's a nice sort of foveal correlation here in the STS, that corresponds to area MT, and that's surrounded by peripheral representations that had strong correlations. So we took this as evidence that retinotopic, actually that should be hidden, sorry, retinotopic maps are present at birth. Now the earliest we scanned these monkeys was just after 1 week of age, I think around 11 days. But we really think that these maps are there at birth. They're not emerging over that first week. But this is still well before face patches are emerging and overlaps with the period of time where these monkeys are preferentially looking at faces. So we putting these studies together, we proposed a framework for how face patches and probably high-level visual cortex develops, where early on there's what we're calling a proto-architecture in the visual system that's comprised of retinotopic maps that already imposes you could say, a scaffolding of the visual system, an organization of how information is getting selectively routed to different parts of cortex. And that's interacting with early visual experience. And in the case of the development of face patches, monkeys are preferentially looking at faces. And so that is interacting with this proto-architecture such that faces are getting selectively routed to foveal parts of cortex. And then over the course of early development, the foveal regions of IT cortex refine their selectivity to become preferentially responsive to faces more than other complex shapes. And we think that this framework is generalizable to all cortical development, such that there are some general organizing principles, some early scaffolding that's interacting with species-specific and individual experience that's leading to the emergence of these specialized regions that are critical for primate behavior. And the interaction of this early proto-architecture with experience can account for the stereotype location of these specialized regions without an explicit predetermination of their function. So more recent work, we've been exploring this idea more thoroughly, and in particular testing how general these organizing principles are. So is there other proto-architecture beyond the visual system? And more specifically, we were asking, well, in other sensory systems, do they have topographic maps at birth? So we've looked at this in the somato-motor system, and that's because in the adult, it's well established that there's a coarse map of the body. That map is topographically organized that neighboring parts of the body tend to be mapped onto neighboring parts of the cortical surface. So there's a region that represents the face, a region that represents the hand and the arm, and near that is a region that represents the trunk, the leg, and the foot. And so we asked whether we could find this organization in our newborn monkeys. So we put our monkeys in the scanner, again, just after 1 week of age, and stimulated their face, their hands, and their feet. And sure enough, we found a systematic topographic organization as early as 11 days. So it was the first scan session that we did. And the organization of these representations of face, hands, and legs and the extent of this coverage matched older monkeys. And this is not just cortically. We saw all the subcortical areas such as the putamen, the globus pallidus, and area VP of the thalamus that all had somatomotor maps. And that was already at 11 days of age. So this indicates to us that body maps are indeed present and pretty much adult-like. We couldn't find really any differences between the maps at 11 days of age and these older monkeys. So these are present at birth. Sort of led us to ponder, though, okay, these maps are there, but what are they good for? I was just talking about how retinotopic maps are providing the scaffolding for the development of face selective clusters in IT. What are these body maps doing? And so we have no data on this, and it's a little speculative, but I think we can have some good estimate of what is going on based on what we know about the adult macaque. So in the adult, there are these motor domains, or sometimes referred to as action domains, that if the neurons in those regions are electrically stimulated, so if you lower an electrode into those regions and you induce some current, the monkey will exhibit these stereotyped, highly complex and coordinated movements. And these are reproducible across a whole bunch of individual monkeys. So for example, if you stimulate this region, this monkey will start to make a climbing, leaping motion. If you stimulate this neighboring region, a monkey will reach out and try to grasp something, even though there's no object in its space. Other regions will evoke chewing and licking action movements. And what's been noted in adults is that there's a correspondence between these domains and the coarse somatomotor map, such that the region that evoke climbing and leaping actions, which utilize hands and feet, overlap with parts of the somatomotor map that represent the feet and the hands. Similarly, the region that evokes a reaching, grasping overlaps with the hand region, and the region that evokes chewing and licking overlaps with the face region of the somatomotor map. So we don't know if these domains are present at birth, but we do know that monkeys lack, like human babies, fine motor skills at birth. They're terribly uncoordinated. So we think this fits nicely with this framework of cortical development, where early on there are these topographic maps that are setting up the infrastructure and scaffolding for how experience interacts with this intrinsic wiring to promote over early development, refinement of the circuitry that gives rise to these specializations that are really important for our behavior and really for our survival. So it remains to be seen when these action domains emerge. But I would speculate that they emerge on a timescale that matches with the refinement of motor skills in monkeys that's highly stereotyped across individual monkeys. And this really underscores an important concept that these maps constrain development. So I'm just showing you here the composite of the results from 2 of the studies of the visual and somatomotor mapping. I'm highlighting a region over here that's auditory cortex. We haven't done auditory mapping in our newborn monkeys, but I highly expect that tonotopic maps are here at birth as well. And I think you can appreciate from this composite image that the majority of the cortical surface has topographic maps, and these are there day one. So there's a lot of infrastructure and scaffolding that is there from the get-go to support and constrain postnatal development. And these maps are additionally important because they provide a bridge across primate species, and I'd argue across all mammals. So across primate species, you can appreciate that the size, shape, and folding varies considerably across individuals. Yet, if you consider the retinotopic organization of these different primates, there are several parallels across these species. For example, each of these primates have a primary visual area V1, a visual area V2, something that could be called a visual area V3 or an area MT, and the location of these retinotopic areas are similar relative to each other across species. So I think that this really suggests that this proto-architecture is evolutionarily old. There are probably species-specific adaptations, but this is something fundamental to cortex by and large, not just probably across primates, but across all mammals. So we've also have been doing more studies looking at what types of species-specific and individual experience is necessary for this early specialization of the visual system. And this is partly motivated thinking about how different our early visual experience is from how we typically test selectivity in the functions of brain regions in the laboratory. So often we'll present images like this to monkeys, and then we'll categorize the neurons based on their response properties. But this is obviously not how we experience these individual objects. They're rather embedded within these rich, complex scenes. And we think that these rich environments are critical for how the brain is wiring up very early on. And these scenes, so these images are embedded not just in these scenes, but objects are sort of experienced with some regularity relative to each other. And there's a nice literature talking about this, that there's a regularity with which objects are co-occurred together in particular scenes. And there's also a spatial regularity to this. And I think this is nicely highlighted in this recent review by Kaiser and colleagues. This is something that is obviously super intuitive, but I think makes the point that there is not just a regularity with which we experience certain objects in scenes, but there's a spatial relationship that is very common, such as lamps are typically experienced above tables, chairs are usually experienced on the same plane as tables, and carpets are experienced below tables, right? And these regularities seem to have some behavioral relevance. So this is some work done by the same group where subjects were asked to identify targets within an array of objects that either had consistent spatial arrangements as what would be typically experienced, or irregular spatial arrangements. And subjects were more accurate at identifying targets in typical spatial arrangements. So this really, I think, highlights that the manner in which we experience objects in our environment influence our behavior. And I think they certainly influence how the brain is wiring up. So this has really led us to start thinking about how the neural tuning and the response parties of IT cortex are reflecting the regularities of our visual experience, not just, is this neuron signaling a face is there? So to unpack this a little bit further, I'm showing you this image of Peter again, playing with one of our monkeys. We have no trouble intuiting that there is a face behind this mask, right? And we can probably do a pretty good job guessing where the eyeballs are, where Peter's mouth is. And this is based off of our extensive experience with faces. And we're using specifically probably this body as a contextual cue of where the face ought to be. Now, on the other hand, Nina here, who has been raised without seeing faces, would probably have a very difficult time inferring what's underneath this mask, because she's never seen a face other than in these experiments where we present faces separate from the bodies. So we wanted to see whether the neurons in IT cortex would be sensitive to this contextual information. So we recorded from face patches in our monkeys while they looked at natural scenes. And we really cared about the spatial information in these natural scenes. So we trained monkeys to fixate. We localized to their receptive fields of the neurons in those arrays. And then we presented these complex scenes at different spatial locations on the computer monitor. And that has the effect of, at different trials, presenting different parts of the image in the receptive field. And we can do this systematically such that we sample a bunch of different parts of the image. So each black dot at some trial, that part of the image was centered on the receptive field of these arrays. And then across trials, we can stitch together all this data that we've recorded and put together these response activity maps. And the way to understand these maps is looking over here in this red region that denotes high firing rate. That indicates that when this face was centered within the receptive field, there was high firing. That's in contrast to when that monkey's hand was centered on the receptive field, where firing didn't significantly differ from baseline activity. So we tested this across a whole bunch of natural images with faces in 2 monkeys. One monkey had 2 arrays, one in the posterior face patch and one in the middle lateral face patch. And another monkey had an array just in the middle lateral face patch. And you can see in each of these images, activity was strongest when the faces were centered within the receptive field. And I think this highlights something that often is not emphasized, sort of skirted under the rug when we talk about ventral visual processing and object processing, and this intrinsic relationship between spatial coding and object processing. And this is critical to our proposal for how these regions are refining and developing their tuning very early on. That there's this intrinsic relationship between spatial coding and this sort of category selectivity or selectivity for complex shapes. So next we tested effects of context by showing occluded and unoccluded versions of images. So here the faces are present and here the faces aren't present, but we could probably infer based on our experience, based on where the bodies are, where the faces ought to be. And sure enough, in the middle face patch of both of the monkeys, we see activity, strong activity when the faces are centered on the receptive field, as well as parts of the occluded images where the faces ought to be are centered on the receptive field. We saw this for all the occluded images we tested. We also saw this for masked images. So these are, again, the masks that we wore while we were rearing monkeys. I should note that these monkeys are control monkeys. They weren't raised without seeing faces first year of life. They did see us in the lab wearing these masks quite often, though. But it wasn't that experience with these particular masks that seemed to be driving these responses, as other images of masked people that they've never experienced with masks they've never experienced also drive these neurons. So this contextual relationship is at a general level of sort of the relationship of these properties of bodies to something above the body and not the specific features. And this effect held even if we remove the faces entirely from these images. So we use mannequins, or we even just cut out the faces and we cut out the background. And just having a body below the receptive field drove these neurons. So it really establishes that the body is providing a strong contextual cue that's sufficient for driving firing in these neurons. And this facilitation seems to be viewpoint specific, and specifically the types of the viewpoints that we typically experience in our faces and bodies in our environment. So we replaced faces with Gaussian noise, so there's no structure to the faces, and we recorded activity within these noise regions when in upright and inverted bodies. And when the body was upright, the neurons fired a lot, and when the body was inverted, the neurons fired very little. And this is the same exact image, it's just rotated, right? So this facilitation of the body in the absence of a face is strongest in upright configurations. So this led us to think about and refine our proposal for this effective early development of preferentially looking at faces being mapped onto a retinotopic map. Specifically, we shouldn't just consider what the monkey is fixating on, but how that's interacting with the regularities of our environment. So in the case of fixating on faces, well, given that bodies are regularly right below faces, then bodies themselves are falling on the retinotopic map in some regularity. And I think that's then no coincidence that we observe in both humans and monkeys face selective regions in adjacent parts of IT cortex to body selective regions. And looking at the retinotopic map, the lateral part of IT has foveal representations. As you move more into the fundus where bodies are located, you see that there's more of a representation of parafoveal space there. So we think when in considering the interaction between early visual experience and this intrinsic architecture, to consider how the regularities with which we're experiencing our environment, not just these sort of isolated experiences. So the lab's current direction is really trying to understand at a deeper level the development of object recognition, the core properties that are associated with primate vision. And one of those properties that's been noted in neurons in IT cortex is that they're robust they have a high degree of tolerability to different viewpoints, recognizing faces, for example, at different viewpoints, under occlusion, like I just talked about, or in noisy environments, or where there's complex backgrounds. And I think that the robustness of our visual behavior and the selectivity of these neurons to accomplish this very complex visual task is what these clusters are particularly optimized for, and probably reflect in sort of the typical adult. And there hasn't been much empirical work on that. There's been some recent computational and theoretical work arguing that may be the function of clusters, or what clusters are demonstrating is a refinement of that circuitry to really efficiently and robustly detect objects in our environment under a whole variety of conditions. And so that's a future direction I plan to go in with the lab. One of the challenges for doing this, though, with macaque monkeys is that you really need to test varying degrees of timing of experience and types of experience. So that doesn't lend itself well to macaque studies where Ns of 2 and 3 are quite common, especially in development where gestation takes about 165 days to raise a monkey. So that leads me to the lab's new direction of using the tree shrew as a model for visual development. And these guys are particularly attractive as a model for visual development because their gestation is much shorter. You can have much larger colonies, so you can have ends needed to really tackle these fundamental questions. And even though these guys aren't primates, they have a architecture of their visual system that's primate-like. They have several visual areas. A large part of their cortex is dedicated to visual processing. And the pathways from the retina to visual cortex have strong parallels to the primate. So there's a primary pathway from the retina to a 6-layered LGN that relays information to V1, consistent with primates. There's a secondary pathway that goes through the colliculus and the pulvinar that feeds into higher order visual areas, just like a primate. And there is natural clustering in the cortex of visual cortex of tree shrews, such as retinotopic maps, and even at a finer scale, clustering such as pinwheel architecture. It's a primary feature of found in primate visual cortex. So I think together this really illustrates that there's probably a similar proto-architecture to the tree shrew analogous to primates. And I think that makes them well suited for studying influences, effects of early visual experience on the development of object recognition. So we've started to explore this by first asking, well, what do tree shrews see? We know that their acuity is not as good as primates, and so we wanted to first see, well, what can they actually see? So we've been running these tree shrews on visual match to sample tests. So they see a cue, and then they are trained to go to the side of the screen that has the matching image, and using natural images. And I have to say, this is special thanks to the Rust Lab, especially Travis Meyer and Barnes Januzzi, that set this up initially, and that my lab largely inherited from their initial efforts in the past couple of years. And I can show you the tree shrews actually are pretty good at this. They love doing this task, and they can do this very quickly. So they go to the center, and then they go to the sides. And so they do this very nicely for these natural images, which suggests that they actually see quite a good degree of detail. However, these natural images vary on both low-level and high-level features. So we've been diving more into what exactly they see in these complex scenes. So there's some, sorry about that. There is already some literature on what their visual acuity is, mapping out their contrast sensitivity. So here's tree shrews, and they see spatial frequency sort of optimally around maybe 1 cycles per degree, which is substantially lower than humans and macaque monkeys. And these studies were done largely before computers in apparatus like this, where there is a nice grading and a mean luminance match grading, and the tree shrews are trained to go to the grading. And so from this, studies have pulled out some measures of acuity. We want to see, using more maybe rigorous methods, what their acuity is. And I'd say roughly we're seeing something pretty analogous to this. So we measured a whole bunch of spatial frequencies at different image contrasts, and they seem to be best around 1 cycles per degree, or just a little bit lower to that. So they indeed have a coarser visual acuity than primates. But that sort of made us wonder, well, what exactly are they seeing in objects? Can they, and what are the stuff that they are seeing, can they really use that in something that would be analogous to object recognition as we think about it in primates? And so that leads to some of the more recent work that we've been doing. This has really been spearheaded by my terrific RA Marcelina, where we are adapting a visual task that has been shown in humans and monkeys to have, so actually humans and monkeys in that first slide in the talk I showed you, the data came from the study, where the ability to recognize different shapes, complex shapes like animals across different rotations, that performance is similar in both humans and macaque monkeys. And so we wanted to see whether such a paradigm could be adapted to tree shrews. So we're just starting out with this. This is very preliminary. And we're starting out without a background. We just want to see, well, could these tree shrews learn to signal, hey, I see this rotated object I'm going to correctly identify the upright version of that object. And we did a little bit of manipulation where we have objects that are highly familiar to the tree shrews, like highly unfamiliar, like pineapple trees, or familiar, such as other tree shrews. And this is really super preliminary data, but we've tested a whole bunch of different objects on 2 tree shrews. Chance is 50% over here. These are the mean average performances across a whole bunch of sessions. The error bars are variance across sessions. You can see one tree shrew for several different types of images, pretty good. Another tree shrew is a little iffy, just skirting like impressively consistently right above chance, but just above chance. But one thing we notice is they tend to actually do pretty well on faces, human faces, monkey faces, tree shrew faces. And if we break these down to images that they have features that they're familiar with, that they experience in their environment, and features, objects that they're typically unfamiliar with, we see some interesting similarity in their performance. First, there's a correlation, there seems to be a pretty good correlation in just these 22 images in performance between the 2 tree shrews. And that's despite a very large difference in their overall performance. So even though Riker is not that great and he's just above chance, that variability across images is correlated with Seymour's performance, who's doing much better. And they tend to be doing much better in performance for familiar versus unfamiliar images. So this gives us hope that, and this is across, they're doing a rotation test. So this is giving us hope that we can scale this up to do something much more closer to what has been argued as a task for measuring core object recognition of primates. And going forward, we also are working on using Penn's fancy 9.4T high field scanner to do functional neuroimaging in these tree shrews. So this is an example of some ex vivo imaging of a tree shrew where we have nice coils to image the top of the brain, but we don't have great signal in temporal cortex. And so right now we're spending considerable efforts trying to build new coils where we get great signal in the regions where we hope to find sensitivity to more complex object shapes. All right, and then on that note, I'd like to thank my current lab, collaborators, and just note I'm looking for grad students and postdocs. And so if anyone is interested or knows people are interested, please get in touch. All right, thank you.
So I'll clap for everyone. That was a great talk, Mike. We already have some questions, and I think let's just jump right in. The first question is from Lisa Marachi, and she says, thanks for the super interesting and informative talk. I wonder if you can say more about proto-architecture and how it might constrain development. For example, you have information about the extent to which face recognition areas in infant macaques not exposed to faces are or can be recruited for other perceptual or cognitive tasks. This question comes up in a couple of the different, this piece of information comes up in a couple of the different questions, like what if they're not faces, what are the neurons in those regions responding to in the animals who don't have face experience?
Yeah, it's a great question. So we haven't quite resolved this. We've been trying to, so I haven't talked about some of these studies, but we're trying to get at this. I mean, the fundamental question is that bit of IT cortex, even if it's not predetermined to be face selective, could it be something else? Could it be repurposed in a way to subserve other functions? And so, let's see, is there a way to easily, I'm just going to do this. Oh shoot, that destroys the screen. Is there a way for me to share my, let's see. Okay, here we go, 1 second. Okay, I just want to go back to an earlier slide. Where are we? Yeah, so I noted, can everyone see my screen?
Yep.
Yeah.
Yes.
So I noted earlier on that these monkeys raised without seeing faces have similar hand regions. What we noticed in these hand regions, they're more extensive, they're way more robust session to session than the control monkeys, which is already interesting to note because typically in control monkeys, the face clusters are like the dominant thing. That's why we study. It's so easy to localize these areas. And the hands are pretty robust, but they can be a little variable. In the face-deprived monkeys, the hand regions are just so dominant. It was only 3 monkeys in this study. We've since run 3 other monkeys and have replicated this, but in those 3 monkeys, it wasn't the size of these patches weren't significantly different, but what they seemed to do was extend more laterally into the region where face clusters tend to develop. And I like to think of this, it's a little speculative, but I like to think of this maybe analogous to these classic studies like the silver spring monkeys, the reorganization of the Somato-motor system, where you don't have a shift of an area, but you have an expansion of the area into surrounding areas where there's a sort of lesion of the sensory input, right? And that's probably constrained by the length of lateral connections. So probably we only would expect this expansion to happen on the order of a couple millimeters. So that's one thing that sort of addresses how flexible IT is. The other thing is, I think to the proto-architecture, I think it's imposing a lot of constraints early on, such that you wouldn't get refinement for selectivities in foveal regions for things that are not typically experienced in the fovea. So if normal monkeys aren't typically foveating on the hands, then those are probably not going to emerge in foveal location. So you have to drastically change their visual experience. And so we are running some studies. I'm running some studies with Marge to look at this, where we're not trying to change the proto-architecture, because that's really hard to, you probably have to do like a Montgomery experiment and mess with the thalamic axons and what they innervate. So instead, we're trying to drastically change the visual experience. And we're doing this in sort of show you a cute little image of monkeys. Where is this? Yeah. So we developed these helmets that we can that these monkeys can raise. And we have cameras so we can record their visual experience, and they have little slots for lenses that we can do things like invert their visual field or drastically change where in the visual field feature, things like faces are falling with respect to the retinotopic map. And right now we have 2 monkeys in this experiment. We don't have any data on it just yet. But the idea is, if we fundamentally change the mapping of experience to this proto-architecture, we might actually see a shifting around of these domains. Okay. So that was a pretty long explanation.
Yeah. Well, I think the next question is on the same theme. From Ling Chi, and she says, "Thanks for the great talk. You showed that retinotopy at birth, together with visual experience, can explain the face selective area. Does this extend to explain the development of other areas, like the place area? Right. Is there a stereotypical spatial, given the stereotypical spatial relationship between different objects? I'm wondering if this is reflected in the spatial organization of all of object selective visual cortex."
Right. So the first thing to note is that, there are scene selective areas. There are strong parallels between humans and monkeys. So this blue area over here is a scene selective region. It overlaps with 2 retinotopic maps that have similar organization across species. So there's this nice relationship with retinotopy again. And these are retinotopic areas that preferentially respond to peripheral stimulation as compared to the face areas that respond to foveal stimulation. So there's this nice thematic link between retinotopy and these functional specializations beyond faces. We haven't done systematic studies looking at sort of the emergence of these selective clusters, and that largely is because of the stimulus that we need to do to cleanly map this region is a little bit different than the stimuli that we used in the early development stuff. And so I'll just sort of go to wherever that is. So if we just take a scene and contrast that with a face here, the scenes span a whole lot more visual space. So what we actually see is the scenes dominate a lot of the visual system. So to really localize that scene selective area, we have to use mosaics of faces and mosaics of objects to sort of roughly balance out the retinotopic stimulation. And because we really want to target face selective regions, we want to just have isolated objects. So we didn't run a sort of second series of these studies with scanning baby monkeys every week for the first 2 years of life, specifically targeting the scene area. We do see, and I might have a slide on this actually, that the scene areas are there in the face-deprived monkeys. I don't know, they exist someplace, but and there, we can identify them definitely around 200 days of age. But before then, we don't have a great marker of like when they emerge. That being said, I'm not sure that we should be drawing direct parallels as far as the idea of the clustering and sort of functional specializations within scene areas compared to sort of the computations that are going on between scene areas and face patch areas. I think they may be fundamentally different, and that's largely dictated by the pathways from early visual areas to IT cortex. And there's been some nice work on this. There's a nice review paper a couple of years ago, really highlighting that these pathways differ substantially beyond just simple retinotopic coding. So, yeah. So no definitive answer, but I suspect that they
But then I guess that sets up an interesting question about the experiment that you're doing now to sort of alter where things are with respect to retinotopy. You know,
yeah.
how much do you think will shift, and how much do you stay the same?
So we did. I hated this experiment. I thought it was doomed to fail, and Marge wanted to run it anyway. We did an earlier version where we raised monkeys, where we plastered faces all over their cage, so that no matter where they looked, faces were in their periphery. And I thought this was like, I don't know, I thought this sort of doomed to work for many reasons, and we failed at moving the face patches into the periphery. What we did see is actually there was much larger face patches in these monkeys. So maybe the wider retinotopic experience was having an effect. But I think, you know, going to this idea of the ability to recognize objects across viewpoints and variants and sort of holistic processing, I think that highlights a circuitry refinement within these regions that probably is different from scene selective areas. And it's not clear to me that these sort of more peripheral regions could wire up to support these similar computations. Maybe, but I think we would need a much better control where we are presenting faces in the periphery and systematically avoiding foveal stimulation. And actually, we are working on a setup where we've adapted these helmets to have a sort of heads-up display, so an augmented reality, where we can use the camera to record the visual experience, and then using a tiny microcomputer manipulate the environment. We've only, sort of doing this first to manipulate things like spatial frequency and contrast, but the goal would be to like do things like shift around faces. But so the first stuff we're doing is sort of trying to shift around the systematic relationships of things like faces relative to bodies, because I think that's the sort of most tractable thing to achieve by just simply inverting the visual field.
Okay. And so the next question, and maybe we'll end on this one, given the time, is about again about plasticity, especially in the animals who have been deprived. So is there any indication of whether face patches can develop after your initial period of deprivation, or are they always compromised even after that experience? This, again, is sort of related to what might be going on in those face patches in the absence of experience. But I think the question is, I don't know, in that experiment, do you then take off the masks and let everybody see your this
was a little bit of a disappointment where
do the face patches come back?
Right. So there's a little bit of a disappointment where we took off we took off our helmets. They started seeing our faces. They started seeing the other monkeys in the colony room, and so we had 3 monkeys. One of the monkeys developed weak face patches. The other 2 monkeys didn't develop face patches. And so that was like not a clean result. I think that sort of probably indicates my sort of interpretation of that is that it's not impossible. So these monkeys started to also look at faces, but not nearly to the same degree as the control monkeys. And so it's unclear whether they didn't develop because they weren't really looking at faces with the frequency with which is required, or was there something fundamentally that has changed about the circuitry? You know, that there's all this experience that's already changing and refining the wiring of IT cortex, and that's now imposing additional constraints. It's making it just that much harder for changes to occur at a year. And that may be a fundamental part of the brain and changes in plasticity, particularly with critical periods, is that once you hit go at birth, there's all this experience that's affecting the brain, and that's having effects down the line of what can and may probably can't develop. And I think the answer is probably it's a mixture of both of those things. And we don't have a good grasp of that just yet. But that's, again, something really hard to do with macaques with ends of 2, but are much more well suited for animals such as true shrews where you can have large ends.
So maybe that answer is to come.
Yes.
Well, I think we'll wrap up there. Heather has popped back in as we cross 1 o'clock. And thanks again for your talk, Mike.
Thanks, Mike.
Great.
Thank you
for being with us today.
Yeah, great.
So just for the closing items of business, I just wanted to say this talk is recorded. It'll be available shortly after we stop filming. You're welcome to join us back here always, of course. Next Friday at noon, we'll have Alexandra Rosati from Michigan, who will be speaking as part of our regular seminar series. And I also wanted to just note out for the MindCORE community that new MindCORE faculty member Nicole Holiday in the linguistics department is giving an ILS seminar in 26 minutes. And she's going to be speaking on intonational variation and incrementality and listener judgments of ethnicity. And so you are welcome to go and hear that talk, and I'll post the Zoom link in chat. So thank you again. Everybody have a good weekend, and we'll see you back here next Friday. Thanks again, Mike and Joe.
Thanks. That was fun.